Hi, Isaac. Thanks for joining me today.
Yeah, thanks for having me.
What inspired your work on the dark kinome? And I think a good starting place is if you could talk about what is even meant by dark and what's a kinome.
Yeah, so I guess before tackling what dark kinases are, it's useful to ask what is a kinase. So kinases are these enzymes that transfer a phosphate group from ATP onto another molecule, usually another protein. And this process is called phosphorylation. It can change the activity, localization, and physical interactions of proteins. So the important thing is that phosphorylation is both rapid and reversible, meaning that proteins can be dynamically switched on or off or between different functional states. So, compared to regulation that occurs at the transcriptional level, this means that these changes occur faster and at a lower cost to the cell. Cells don't have to destroy or create new proteins in order to regulate them. And so, because of this, kinases play a big role in regulating just about every biological process you could think of. And in humans, there are well over 500 kinases, but a substantial portion of them, maybe about a third, are considered under researched or dark, meaning we don't know what they do. So, several years ago, the NIH had this Illuminating the Druggable Genome initiative and defined a list of these kinases. I think there were about 162 of them on this list. And they defined that based on metrics of number of papers mentioning these proteins, small molecule inhibitors, patents mentioning them. And so, supported by that funding, we've been one of the groups trying to generate essentially just some basic information on them with the idea that this can create a starting point for other scientists, other groups to follow up and pursue some of these kinases for further study.
Yeah, I think that's a great explanation of the darkness part of it really stood out, but it must be really interesting to be on the cutting edge of this work in terms of looking at things that really haven't been focused on by a lot of other people.
Yeah, it's exciting because, well, one, because it's unknown. I know that being in the unknown is just an exciting kind of place to be. One of the things I like about it is there's no specific disease angle. And so some of these kinases are related to neurological neurodegeneration, some are related to growth signaling, cancer, others are related to other areas of biology that I've never really heard of before. So it's fun as a researcher getting to see all these different areas and collaborate and interact with a variety of people on these projects.
And just because it isn't tied to anything, that doesn't mean it's not significant, right?
Mhm.
So, how does it help our understanding of cell regulation and potential therapeutic development?
Yeah, so I mean, the easier part of that to answer is the therapeutic development. Kinase is considered one of the most druggable classes of proteins. They have this ATP binding domain that was required for their catalytic activity. And so it's fairly straightforward to design inhibitors for a kinase by blocking that binding domain. And so there's already a number of FDA approved small molecule inhibitors for kinases, probably more than half of them for cancer therapies, just because those signaling pathways have been well studied and kinases have there's a handful of well studied kinases that have very established roles in those pathways. Because of the under research, the dark kinases are seen as having a high therapeutic potential. But before you can choose those as a target for a new drug, you need to understand what types of disease pathologies they might be relevant to. That's one angle, the potential of these proteins as drug targets. And I think the other kind of more interesting angle is just like the phosphoproteome at large. There's on all the different proteins in the human proteome, we've detected hundreds of thousands of phosphorylation sites on all these proteins. Maybe about half of those phosphorylation sites have distinct effects on the protein's function. But the problem is that most of those sites are not mapped to an upstream kinase. So if you're studying your favorite protein, chances are it's phosphorylated and it changes its function, but you likely don't know what upstream kinase is responsible for that. About 95% of these phosphorylation sites have no known kinase, and those that do are mapped to this small subset of well-studied kinases, usually related to growth factor signaling. The other thing that's interesting about this is we can level the playing field and get a more unbiased mapping of the phosphoproteome to allow us to better understand disease pathologies and things like that.
You may have noticed that a lot of my questions focused on your application of mass spectrometry. Certainly, that and chromatography are the two main focuses of this podcast. So, I was curious if you could describe the specific mass spec workflows you used and what was the rationale for employing each of those in this study.
Yeah, our lab's technology of choice, at least in the initial stages of this project, has been proximity labeling, which is this technique where you can take a protein of interest and tag it with we call a promiscuous biotin ligase. So this extra enzyme that we attach to a kinase of interest will label nearby proteins in the cell when you add biotin to the cell. This allows us to label nearby proteins and see the kind of cellular neighborhood around one of these dark kinases. And so that tells us what organelle it's in, what protein complex is it part of, and it also kind of gives you a list of all of the proteins that are kind of within the catalytic range of the kinase. So all of the set of proteins that a kinase could potentially phosphorylate. The other technique that we've used is, of course, phosphoproteomics, where we can enrich for all of the phosphopeptides in the cell and quantify those. So this allows us to see what downstream phosphorylation changes are occurring when a kinase is genetically perturbed or perturbed with a drug. And so the combination of these two techniques can give us a good idea both, where the kinase is in the cell and then what proteins and pathways it's affecting.
With mass spec, you're generating a lot of extensive and complex workflows, but how do you approach that?
Yeah, the missing data is a kind of big challenge in the proteomics field because it impacts the downstream statistics. If you have missing data that is essentially censored and affects your ability to detect differences between groups, I think it's important to think about the source of the missing data, right? So like one component is missing at random. The process of, if it's DIA searching these complex spectra, or if it's DDA, the process of selecting specific peaks for fragmentation is inherently a little bit random. And so some identifications are missing completely at random. But another component of the missing values is related to the abundance of the peptide you're detecting and the limit of detection of the instrument. So data can be missing also because the quantitative value is critically low, right? And therefore, that type of missing data actually carries meaningful information, right? So the absence or presence of an identification does tell you something about the biology, but it can be difficult to parse out the two sources of randomness. That's kind of one of the challenges I work on. People typically will impute some small value to use as a placeholder and enable statistical tests, but this can be a little bit unpredictable and perhaps risky. If the imputed value is too low, then your stats are inflated, right? Your p-values will be better than they should be, and you'll have a high false positive rate. Whereas if the imputed value is too high, then you maybe don't have the sensitivity to detect the things that you want to detect, the biological signals that you want to detect, and maybe your false negative rate is too high. One approach I've worked on is to use the missing values as a way to inform the value that you impute. If something's missing all the time across many different mass spec runs, across many different types of samples, that probably means the baseline abundance is low and you can choose a low imputed value. Whereas if something is frequently observed, maybe you want to choose a higher imputed value. So this is the type of thing, specifically with proximity labeling data, where the presence versus absence of a protein is informative, incorporating that into our kind of data analysis approach.
Absolutely. Looking into the future, how do you envision advances in mass spec impacting the protein to protein interactions and cell signaling beyond the kinome?
Yeah, I mean, everything is kind of crazy. I mean, I'm now a fifth year grad student, and things have changed so much even since I've been in grad school. It's kind of insane. Nowadays, everything is much higher throughput. The mass spectrometers scan so much faster that you can run so many more samples and also use lower amounts of input sample. And so you can kind of see where this is heading. It's heading towards being able to do proteomics and phosphoproteomics off of single cells or near single cells. This year, the TS Mann's lab had a preprint on their nanofos workflow, and it's getting essentially very close to being able to do phosphoproteomics analysis off of single cells. And that's, I think, going to be game changing for systems biologists because it allows you to dissect out the heterogeneity of tissues. I think, for example, if you're studying mitosis or a process related to mitosis and you have a tissue sample from a mouse or a person, a very small percentage of the cells in that sample will be in each stage of mitosis. And so when you do a bulk proteomics analysis, what you're getting is essentially the average. And so you may not be able to detect differences in any particular stage. But if you can dissect out or parse out the different types of cells or look at things at the single cell level, you might be able to see these changes that are distinct at the cellular level but hard to detect at the tissue level. So I think this is going to allow us to see a whole bunch of processes in more detail that are difficult to study right now.
In terms of the high throughput screening, could you talk a little bit about both TMT and DIA and how that assists with this analysis?
Yeah, so these are the two different mass spec data acquisition techniques that we're currently using for phosphoproteomics. The TMT labeling is a strategy to where you can label different samples and then combine them, multiplex them. And this has a couple of advantages that kind of give you unbeatable data quality with the TMT labeling. You get higher depth because you're multiplexing. You can afford to use offline fractionation and divide the sample up and get higher depth. And then you also get this improved quantitative accuracy with the TMT reagents. And then the other huge advantage, particularly for phosphoproteomics, is consistency. Each time you do a phospho enrichment, these are usually B-based enrichments, there's variability in each of those enrichments. And so when you multiplex, every sample is essentially part of the same enrichment, and you don't get sample to sample variability. And so for all of those reasons, the TMT labeling is kind of superior data. The other big thing is the missing values. You don't get missing values with TMT, whereas you do with DIA. And so that's a huge advantage. The problem with TMT is that it's a much more laborious workflow. It's just a little bit more of a pain to prep those samples, and it's more expensive. Often we'll use DIA as a kind of budget, high throughput, quick way to get a phosphoproteome, and then if we want, we'll follow up with TMT. So DIA, we're now running samples out of 12 well plates. We have this Kingfisher robot that'll do the magnetic bead preparation, phospho enrichments for us. And so we can go from a plate of cells to samples running on the mass spec in as little as 48 hours. And so it's just allowing us to test more conditions, see what types of experiments on the biology side, what transfections, what drug treatments work before we go in with the expensive TMT labeling approach. The number of conditions. With TMT labeling, you have essentially a max number of samples that you can get in an experiment depending on the I guess the plex of the reagents. Whereas with DIA, you can get as many conditions as you want, and you can also add conditions to the data set later, as long as the chromatography and everything is somewhat consistent. So this allows you to collect bigger data sets with more conditions, which can have advantages for data analysis.
You touched on how much things have changed or improved since you started this work. In that line of thinking, is there anything specific that stands out there where you could think back and say, oh, this is totally different now compared to how we used to do it?
Yeah, I mean, for sure, the way we're doing phospho-enrichments now on the robot versus me doing it by hand with a magnetic tube rack is very different. Before I could do a phospho-enrichment in maybe 2 hours on 6 samples, and now the robot could do 96 in 1 hour. So it just means we can have a much higher throughput and do more samples. And then the other big change is DIA. When I started, everything was being done DDA, and now the field has made this big shift towards DIA data. And so that's maybe the other big change.
I just knew that the robot was going to be part of that answer.
Yeah, it makes it just so much easier, and it's consistent too. When I do it by hand and I vortex the beads, if I do it for 5 seconds versus 7 seconds for one tube versus another, that's an inconsistency that can show up in the data. But the robot doesn't do it. Every sample, it's treated pretty much exactly the same.
And that's what you're going for.
Yeah, and yeah, how exactly that shows up in the data analysis can be tricky to tell, but we see lower variance between replicates and phosphopeptide abundance, and then also missing values. Consistency in the phospho enrichment leads to more consistent identification of phosphopeptides. Those two things have a big impact on what you can get out of the data.
I'd be curious to hear what have been some of the more surprising or challenging aspects of this work for you personally.
Yeah, I mean, I think one challenge is, especially with the dark kinome project, is it can be tough to tell when you have good data, especially when you operate kind of way out in unknown with proteins where you don't really know what they're going to do. So we're working, part of it is we're working with somewhat new, recently developed experimental methods. Proximity labeling and phosphoproteomics, they've been around for a while, but they're still somewhat new. And then combining that with under research targets that you don't know what the effect is going to be, it can be difficult to know kind of when an experiment worked. And so despite studying dark kinases, I find myself a lot of the time actually working with the well-studied kinases to optimize experiments, essentially asking like, what for these kinases that already, where you know what they do, what can you feasibly learn from these types of experiments, and doing that first before going to study the dark kinases. So that's been a little bit surprising. And even with the well-studied kinases, it's challenging. They're sensitive biological systems, and there's variability in the sample prep and the technical aspects of the data acquisition. So all these things combined can make it kind of challenging to get good data and then know when you have good data.
The audience for this podcast is young scientists, and I was interested if you had any advice for them, people who are interested in studying proteomics, using mass spec or systems biology in general.
Yeah, that's a tough question. I almost consider myself still a young scientist. I think this field is like so vast, it feels like at times. It's simultaneously deep and wide because it's like this combination of so many different fields, right? There's the math, there's the physics, biology, biochemistry, organic chemistry, computer science. I think that can be a challenge, but I think it's also very rewarding to be combining all these things together. I think the advice I'd say is don't be intimidated by that. That is partly what makes it fun and challenging. So don't be afraid to ask all sorts of questions, even if you think they're dumb questions, because it's not possible to know everything, and all these things affect your experiments and your data. I'd say lean into this complex aspect of it and have fun. It's some of the most advanced technology on the planet, and getting to work with it is a huge privilege. It's super fun.
I think that's great advice. Um, in some ways, it's turning that feeling of intimidation or being hesitant into just pure excitement, I suppose, and that willingness just to dive in and just turning nervousness into excitement.
Yeah, I,
Was
it's as a kind of rapidly growing field. That's I think it's a fun spot to be, and people innovate off of all these different areas that I just mentioned. And so that also means that there's lots of opportunities to apply your skills. If you're particularly good at any one of those many things, that's an opportunity to innovate and make an impact.
Well, Isaac, I really appreciate the time today. Thanks so much for sharing your work with the audience.
Yeah, thanks. Thanks for having me. Honored to be able to share my research with you.